Hi, my name is Kevin Ahern, and I'm a professor of biochemistry at Oregon State University.
Blood clotting is a pretty essential thing for a multicellular organism. All right? If you think about it, and we sort of take it for granted, but if you think about what has to happen, all right? If I go out and I cut my finger, okay? If I don't have something to stop the blood flow, I will bleed to death. Now, we take it for granted that we don't have to think about that because we have a clotting process that takes care of that. But it's important to remember that. We've got a clotting process that takes care of that. Okay? We can even, repair and stop the blood flow for fairly serious types of cuts that we have. Now, if we think about this for a more serious type cut than, say, cutting my finger, we realize that the challenges that the body has in trying to manage blood flow. All right? So, for example, when I cut, let's say, a major artery, all right, then my body has got to very quickly put something in place to stop that blood from exiting the body and keep it in the system that's there. Okay? It has to have a system that is ready to start literally at the drop of a hat, and it needs to assemble a repair, or at least a temporary repair, within minutes, because if not, you're going to bleed to death. More importantly, that repair system should only be activated by an injury, because we don't want it forming clots when there's not an injury, because clots traveling through our bloodstream hit our brain, hit our heart, cause heart attack, cause stroke. Okay? This is a very big demand that we've placed on our body. It means that we need to have floating in our bloodstream the ability to form a clot very quickly, a hair trigger, but that hair trigger only fires when we want it to fire. Okay? Now, to do this, the cell accomplishes this by using a cascade. Here's what the bloodstream cascade looks like. To show the complexity of what's involved in making a blood clot. The blood clot is formed down here at the bottom. Okay? Here's where the blood clot forms. All right? It forms as a result of the self-assembly of protein subunits called fibrin. Okay? When you have fibrin present, they will self-assemble and they will form a structure that ultimately will hold water. It will be water resistant. Okay? This is nothing short of remarkable. And it does it in seconds or minutes. The active forms are in yellow, the inactive forms are in red. Well, if we start at the very top up here, we see that there's a cascade that happens. We can actually activate the clotting process by two different pathways, and I'm not going to talk about what they are, but two different pathways can lead ultimately to a blood clot. And interestingly, the two pathways converge at a point, meaning I could start over here and make a clot, I could start over here and make a clot, or I could start at both and make a clot. If we look what it is that converts, that makes fibrin, all right? Fibrin starts out as an inactive form. It's called fibrinogen, and our bloodstream is loaded with fibrinogen because we like to be able to make clots as quickly as we can when we need to. Fibrinogen is the inactive form. It's converted into fibrin by action of a protease known as thrombin. And that causes this bottom part of the pathway to occur. Okay? We're going to focus essentially all of our attention down here on this lower part, because for our purposes, this is where the real action is going to be. Okay? So, we're interested then in the conversion of prothrombin to thrombin, fibrinogen to fibrin, and the formation of a blood clot. The blood clot is made initially as a very loose structure, and then we'll see how it gets hardened with action of an enzyme. And you probably have seen this with clotting on your own skin. That the initial thing that will form will be a very loose kind of clot that you can bump and cause it to bleed again very readily, but you give it a couple of hours or you give it a few hours and you get something that's dry and that is much more stable and solid. and that happens as a result of hardening of the clot that we'll talk about. Okay, so let's look and see what's happening here. All right, so we're going to start down talking about fibrinogen. All right, and fibrinogen looks like this. Okay, fibrinogen has 3 chains associated with it. All right, they have an alpha, a beta, and a gamma. 3 chains. We see that it's a dimer. Here's another alpha, beta, and gamma. And we see that the alpha, which is this red guy, has a structure on the end of it called capital A. It's a good name for it, capital A. All right? And we see that the beta, which is shown here in blue, has a structure on it called capital B. Simple to remember. Alpha is A, beta is B. All right? What thrombin is doing is it's clipping off so that the capital A and the capital B disappear. We're left with the little skinny part, but the fat part is disappearing. Okay? Everybody with me? Now, that removal of the capital A and the capital B is sufficient to cause the polymerization to start to occur. Because the A, what's left over of the alpha, where the A was, this little skinny part right here, the skinny part of the alpha, can fit into this part of the gamma. Okay? So we see this little red guy can actually link to one of these, and this can link to another one of these. And we can imagine now that we could make an entire sheet of these by these guys linking inside of that little unit right there. That makes sense? So the alphas are going into the gammas and forming a loose structure. All right? Well, what about the betas? Well, it turns out that the betas can fit into the whole of beta structures. There should be one over here as well, but they can fit into the holes of these beta structures. And now we get a 3-dimensionality of it because it's coming back out at us. So the polymerization that's happening is linking this dimer to another dimer, to another dimer, to another dimer, and it's occurring not only in the plane of the board, but it's also occurring this direction as well. All right? That's happening solely because the A's and the B's got cleaved off. So this structure gets bigger and bigger and bigger. Well, if we think about it, that's what really is happening. If we were to look at any individual protein, that would be any individual dimer that's there, it would be way smaller than we can see even with a microscope, even the most powerful microscope. We wouldn't see an individual one. But this structure can polymerize and grow thousands and thousands and millions of units to the point where we can actually see a scab. We see that clot that forms. That's really remarkable. A really remarkable thing. Okay? Let's look at a polymer. So here's a polymer. You don't have to envision it. You can see now how we have these guys that are sticking together. Okay? There's the A's with the gammas. The ones coming out at you don't see because they can't really draw it in 3 dimensions. But we see all these various holes for the individual betas. The beta blues are going to stick in those and now come out in this direction. Okay? Hardening of the clot occurs through covalent modification. Here comes covalent modification again, guys. You know, what does covalent modification do? It gives you a strong bond. That theme has come through in disulfide bonds. It's come through in several things we've talked about. But that covalent modification gives strength to the clot. Okay? How does it work? Well, there's an enzyme called transglutaminase. And transglutaminase will catalyze a reaction between glutamines and lysines that happen to be close to each other. Okay? Is there a specific one? No. All right? Are there many glutamines and lysines that in this mess of protein that's in this polymer, are there many of them that are close together? The answer is yes, there are. So now we're forming covalent bonds between the individual dimers that are there, and those covalent bonds give this unit strength. So the hardening of the clot is made possible by the action of the enzyme transglutaminase, and it's making this reaction right here. Okay, so that's cool. All right, questions about that? Yes? Okay, good question. How do we keep transglutaminase from acting where we don't want it to act? Well, as we will see, many of these enzymes are localized pretty close to where the clot itself is forming. I'm going to give you a mechanism by which one of those gets localized in just a second. Okay, but good question. We don't want these things going anywhere besides the clot. This question is very good because we don't want all this process happening elsewhere, because now we're going to start taking all of our proteins and putting those together. We don't want that, right?